The Department of Life Sciences was always committed in enhancing the research and practical skills of the students through various internship programs which we conduct every year. So this year, as a part of BioSkill Studio, we are organizing the DNA barcoding and molecular phylogeny internship for the first time from the department. DNA barcoding is a molecular method with which we can identify any living organism. And phylogeny is where we can understand the relationship between different organisms by constructing a phylogenetic tree or a family tree. So here, this internship is designed in such a way that the students will be able to understand DNA barcoding and molecular phylogeny. Along with that, they will master in the molecular biology techniques like DNA isolation, then PCR amplification, primer designing, and also data analysis and interpretation, phylogenetic tree construction, etc.
In this internship program, our students have been exposed to both wet lab as well as dry lab experiments. Under dry lab experiments, they were actually exposed to various tools, especially tools such as BLAST, Primer 3, which is useful for primer designing, as well as they were exposed to molecular phylogenetic tree construction tool called as MEGA. And all these tools actually help them to understand how exactly they are involved in making the phylogenetic tree from the data that is being generated using the DNA barcoding information that they have obtained from the samples. Not only this, the students have also been exposed to DNA sequencing techniques and methods so that they will realize how exactly the DNA which is obtained from the plant samples are being studied till their evolutionary relationship that is existing between different members. In addition to understanding the molecular phylogeny, the students are also being taught about how to submit the sequences that they have generated using their own samples which they have collected from our own campus. Altogether, I should say that the students have understood the basic principles, concepts, as well as the techniques that are required for DNA barcoding, as well as the molecular phylogeny prediction aspects, which involves both wet lab and the dry lab experiments. This will definitely help the students not only to understand how these techniques are playing a very vital role in understanding the organisms, but they also help the students to think beyond these aspects in the upcoming days.